lot of people hate it though. It depends on like how you try. I know. Yeah.
Guys, I'm gonna send in the worksheet. I don't know if it's right.
Look, you do a basket here when this is probably like around right now.
What is this spring break? Oh, you can guess. Yeah, March 10th. Sorry.
You about to get you know, this one, I was like, really sometimes. That was literally during football season. Now tell her to try again.
Oh.
Or look at this. If they listen to it, you can catch up with their football season last year. I'd say.
I was gonna ask for a say if the people who were not gonna be here, if you can take it before the Wednesday.
Yeah.
Because we didn't have that with it.
Yeah.
Because I'm gone those 2 weeks. So yeah, I don't have everything. Okay, thank you.
Oh, here's another one, literally at camp.
Well, I talked to Anna. I thought it was.
Yeah, I thought it was. It's like right next to this is
Oh, really? Yeah. I love her.
I'm not reposting. I know. Oh, so how do you know? When did you send the thing?
That was cheesy, man.
I just sent you what I did.
I saw that.
I feel bad.
Alright, guys. quiet down. Alright, we have a lot to get through today and Friday, so let's get started. Riley? Here. Brianna, A. Drew? Bria? Henry? Anna? Nick? Abby? Here. Hannah? Here. Rosa? Here. Matt? Jillian? Here. Mia? Here. Annie? Brianna? Sadie? Here. William? Marissa? Sophia? Aubrey? Here. Matthias? Here. Colin?
Here.
Olivia? Here. Braden?
Here.
Amber? Here. Katie? Kennedy? Ben?
Here.
Ava?
Here.
Kendall?
Here.
Emily?
Here.
Lauren?
Here.
Jennifer?
Here.
Isabel?
Here.
And Corwin?
Here.
All right, awesome. So, today, well, just a reminder, exam 2 is next Friday. It'll cover 3 chapters, so it'll be less material, but it is more difficult material in my opinion, so I hope that you guys have been studying and the worksheets are helping stay on track. So, we've been talking a lot about bacteria and how they're constantly going into these different environments, and so the bacteria has to know when it needs to alter its physiology, and with that, it has to alter its gene expression profile, meaning it has to change which genes are turning on and turning off. So we call that molecular regulation because bacteria need to be able to turn those genes on and off at the molecular level. So we'll talk about what gene expression is, the different levels of control of how the bacteria can express those genes, control of operons, and then on Friday we'll finish off with quorum sensing. We talked a little bit about that. We'll go into more detail. So an overview, we'll talk about the lactose operon, other systems of operon control. But just to orient you a little bit, a bacterial genome, so all the genes within a bacterial cell, encodes thousands of different proteins for many different environments. Some genes are tailored for specific environments, but not for others necessarily. And so, the microbe isn't going to waste its energy trying to make proteins that are not needed at that point. And so it needs, and this has a lot to do with it being able to successfully compete with other organisms in that niche. So there's these elegant mechanisms that are used to control the expression of their genes. And this can happen at different levels. It can happen at the transcriptional level, where literally transcription is when you go from DNA to mRNA, so you can either stop transcription of that gene or promote it, so you're increasing or decreasing gene expression. These mechanisms can occur at the translational level, where translation can be impacted or the mRNA can be degraded before it can go through translation. And then there are a number of ways in which you can actually modify the peptide or the protein after translation, so post-translation. And so we talked a little bit about, degradation, sequestering certain proteins. But today, what we're going to talk about and focus a little bit more on is this control of transcription. So there's different levels of control, like I said, and so that could be, at the DNA level, at the transcriptional level, which is what we'll talk about today. They can be, certain proteins can be repressors or activators. We'll talk about sigma factors a little bit more, and then there's obviously other levels. You don't need to know all of these different things and what level they're at. We're going to focus on control of transcription, but one thing you should know is the post-translational control, which we already talked about, which is all of these different types of things. You can cleave that N-formal methionine, or you can phosphorylate, methylate, acetylate, just add those groups after translation. But one of the things that you guys should know is that controlling the gene expression at the DNA sequence level is the most drastic and least reversible, because you're literally controlling the DNA sequence. Whereas if you are trying to control expression at the protein level, that can happen more rapidly and most reversibly. Does that make sense? Okay, and that's because at the protein level, the genetic code has been maintained. You're altering the protein, so it's more reversible. So now getting right into it, so a microbe needs to know if it's just floating in a pond or if it's in your gut or wherever it is. So it needs to be able to sense its environment, and so it can then change its gene set that it's expressing to meet the needs of, the survival of in that particular environment. And this is largely controlled by regulatory proteins. Regulatory proteins are what helps the cell sense change, internal change, and alter its gene expression to match. These regulatory proteins have to specifically bind to compounds that are provided by in the environment, which we call ligands. So I tried to keep the colors consistent here, so you can really use the pictures. They're really helpful in trying to keep these things straight. So you have a regulatory protein that will either control, that can control the gene expression. And so regulatory just means that it could regulate it. So it doesn't mean it's going to increase or decrease expression. Regulatory proteins can be either. So this is the general term for a protein that can regulate the genes. And then there are ligands, which are molecules that, or compounds that specifically bind to the protein to help it do what it needs to do. You'll notice here that there's this green regulatory sequence, so just keep in mind that you have RNA polymerase there, you have the promoter, and then the target gene that's going to be transcribed. So the regulatory protein is going to bind to a regulatory sequence that's right before the RNA polymerase, and so that helps regulate gene expression. So it's got to come right before. Does that make sense? The regulatory protein is also encoded on the DNA. It has its own promoter, and so it's its own protein. You have to make RNA, and then you make the protein, so it goes through its own set of transcription and translation. And different regulatory proteins will bind different sets of ligands. Does that make sense so far? So regulatory proteins can come in these two different forms, activators or repressors. Activators bind essentially the regulatory sequence. So if this is an activator protein, it's going to bind that regulatory sequence, and it's going to stimulate transcription of the target gene. Most activators have to bind a small ligand first. There are exceptions to the rule, but most of them have to bind a ligand first. And we'll talk about what some of these ligands look like. Repressors do the opposite. They also bind the regulatory sequence in DNA, but instead of activating transcription, they prevent transcription. some of them bind a small ligand, some don't. So far, it's intuitive, and so now we're gonna just introduce some more terms, some different types of ligands. So most activators bind poorly on their own and they need a ligand, in this case, an inducer ligand. So here we have an inducer ligand, which is the green diamond. It's gonna bind to the activator protein. That then allows the activator protein to bind to that regulatory sequence, and it helps to activate transcription, and the target gene is expressed. Simple. However, like I said, bacteria need to regulate these genes, so there's got to be the other side of the coin available. So if something is able to activate transcription, there's also going to be a mechanism where it can also stop transcription when it's not needed or when you run out of the ligand. So in this case, activation or repression using an activator protein is actually dependent on the concentration of the ligand in the cell. So if you have high levels of the ligand, you're going to have enough of the ligand to bind the activator protein and activate transcription of the target gene. If you have a low level of the inducer, so as it gets used up, you have to remember that these things aren't just there constitutively. You have to, they get used up and then you have to have them come in and out of the cell. And so if there's low levels of the inducer ligand, the activator without the ligand isn't able to bind. It's kicked off, it doesn't bind this regulatory sequence, and so now the target gene is not expressed. So in this particular example, this target gene, in order for it to be expressed and transcription to occur, you need the activator. The activator can't bind on its own, it needs the inducer ligand, but like I said, when that runs out, then there's always that other side of the coin where, the target gene isn't expressed. They have to be able to go back and forth. Does that make sense so far? Any questions there? So now let's talk about a repressor protein. There are different typical scenarios that can happen. And so there's two general ways depending on the mechanism of the repressor. The repressor, hence the name, will repress the target gene expression. So the repressor binds to the regulatory sequence on its own, and the target gene is not expressed. It's what we would expect. But like I said, there's got to be a scenario where you have relief from repression. And so this requires the ligand, also an inducer in this scenario, where this time the ligand binds to the repressor, and then the repressor falls off of the regulatory sequence because now the ligand binds it, and now the target gene can be expressed. So the inducer binds the repressor protein, causing it to release from the DNA. So now I've told you that the inducer ligand can do two things. It can bind to the activator and allow the activator to bind to the regulatory sequence and allow transcription. And the inducer ligand can also bind to the repressor, but instead of letting it bind, it falls off and it activates or induces transcription. So in this scenario, the repressor protein is able to bind the DNA on its own. Remember I said that some of them need a ligand to bind, some of them don't. In this scenario, the repressor does not need its own inducer ligand to bind. The ligand rather is an inducer, and it induces transcription because it releases the repressor protein from the DNA. Does that make sense so far? So in the second scenario, the repressor protein can't bind on its own to repress the expression of the target gene. It actually needs a ligand called a co-repressor. So in scenario 2, the repressor itself binds poorly to the DNA sequence and it needs the co-repressor ligand in order to bind, and therefore the target gene is no longer expressed. But then once again, as the concentration of this ligand starts to disappear from the cell because it's being used up to repress transcription, it eventually becomes no longer available to bind the repressor, and then that original repressor protein dissociates from the DNA, and the target gene is therefore repressed or expressed. And so this is called de-repression. It's not necessarily induction because we're not using an inducer ligand, but it's the removal of the co-repressor and therefore the removal of the actual repressor that allows for de-repression and target gene expression. So a lot of terms I'm throwing at you now, and I know it can get confusing. So try to, think about this and ask questions if you need to, because it can get a little confusing. So now I've told you about two different types of ligands, an inducer ligand and a co-repressor ligand. The inducer ligand has roles in both activator regulatory proteins and repressor proteins. Co-repressors have roles in just repressor proteins. Questions so far?
So in de-repression, that's when the target gene comes back that you were supposed to get rid of in repression? Okay.
Yeah, but there's always the two sides. You always have to have the ability to activate the gene. You're not repressing it forever. It just gets repressed when the environment needs it to get repressed. And usually that's going to be known because the co-repressor is there. There's a lot of it and in the intracellular environment. So we're talking about inside the cell. And so when the co-repressor is there, then it does its job. It binds the repressor. The repressor is able to actually repress transcription and bind to the DNA. Whereas, then it runs out. It's all about concentration, intracellular concentration. You run out of the ligand, the actual repressor falls off, and the target gene that is then starts to get repressed. Any other questions? So once again, this is all intracellular signaling and regulatory mechanisms. So this is all inside the cell. It's dependent on a lot of concentrations. But now, we have to think about the bacterial cells sensing the extracellular environment. And there is a very common mechanism that's used by all bacteria to collect information from outside, and that relies on this two-component signal transduction system. This includes two proteins, a sensor kinase, and a response regulator. Now I'm throwing more terms at you. So remember, these are associated with sensing the external environment. The first protein here is the sensor kinase in the cell membrane. So remember, you think of a bacterial cell. So this is inside the cell where you have the actual genes, and you have the polymerase, the promoter, the gene that's going to be transcribed. And so this is the outside of the cell. And so you have the sensor kinase is in the membrane, embedded in the membrane. So it's a protein with enzymatic activity. And the sensor kinase will sense and detect some environmental signal on the outside of the cell. This signals what is called autophosphorylation, which means that the sensor kinase can activate itself via ATP autophosphorylation. So ATP is phosphorylated in this case. The response regulator is in the cytoplasm inside the cell, in orange. And that then, because of this autophosphorylation, it can then just take that phosphate from ATP. And once that phosphorylation happens, when that response regulator is phosphorylated, then it automatically can bind to the chromosome and it can either alter the transcription of genes. So it could be either an activator or a repressor. So remember, anything that's a regulator just regulates. It can be positive or negative regulation. And in this case, it could be either, but there's this two-component signal transduction pathway for all bacterial cells, where you have a sensor kinase senses an environmental signal. That results in autophosphorylation. That phosphate group gets transferred to the response regulator, which allows it to bind to the sequence and either transcribe or repress transcription. Does that make sense? Okay, quick concept check. Transcription is prevented by blank and stimulated by blank. I know I threw so many words at you, so try to remember what we talked about. The regulatory proteins that either prevent or stimulate transcription. Yeah. E. Transcription is prevented by repressors and stimulated by activators. So those are the regulatory proteins. Remember, inducers and co-repressors are ligands, and regulators regulate either way. So the answer is E. Does that make sense? Okay, one more question. An inducer ligand can bind to what? Both. Does everyone agree? Yep. So let's just make sure that we really grasp that. So an inducer ligand, when it binds to an activator, what does it do to the activator protein?
It binds that to the sequence, then it increases the transcription, and then when it runs out, it just stops.
Exactly. So an inducer ligand, when it's bound to an activator, it allows the activator to bind to the regulatory sequence, and it can then transcribe the DNA. On the other hand, an inducer ligand, when it binds to the repressor, it makes the repressor fall off, and therefore it induces transcription. So there are different roles. When it's bound to an activator, the activator can bind to the DNA and activate transcription. When it's bound to the repressor falls off, and that also induces transcription. That's why it's called an inducer in both scenarios. Does that make sense? Okay. Great. All right. So now let's take some of these principles and apply them to some operons. So Jacques Monod and Francois Jacob proposed that genes could be regulated, and they did this by E. coli, is one of the main bacteria that's studied. They noticed that in E. coli, enzymes that metabolize lactose were inducible, while those that metabolized glucose were constitutive. So lactose and glucose are two different types of sugars, and in lactose, the enzymes that break down lactose in E. coli, you could induce them, but enzymes that break down glucose were constantly being expressed. They were constitutive. And so, can anybody maybe guess why there's the enzymes that are used to break down glucose would be constitutively expressed? They're just always there. Yeah.
For glycolysis?
Yeah, for glycolysis, because glucose is usually the preferred method or the preferred energy source for cells. Also, lactose is broken down into glucose and galactose. So glucose is a monosaccharide, lactose is a disaccharide. So you usually have to break down lactose in order to get glucose. And then glucose will then go into glycolysis to produce energy for the cell. Great. So they recognized that there's this enzyme in E. coli that's used to consume lactose, and it was only produced when lactose was added to the growth medium. So they coined this term induction, and this really helped launch this field of gene regulation. Enzymes for glucose catabolism are always present and produced at high levels because glucose is the favored carbon source, provides the quickest source of energy, and then it can get right into glycolysis. So let's talk a little bit about this lactose operon, how cells utilize or break down lactose. It's this LAC-ZYA operon of E. coli that is really this paradigm that helps describe a lot of these. So you have a cell. This is an E. coli cell. This is the membrane. This is the outside of the cell, inside of the cell. Lactose is a disaccharide, the two sugars that are attached. And there is an enzyme or protein inside of the membrane called LAC-Y. It's a lactose permease, so you guys should know. Remember, this operon is the LAC-ZYA operon. So we're going to talk about the LAC-Z, the LAC-Y, and the LAC-A. The LAC-Y is the permease that allows lactose to permeate inside of the cell. LAC-Z is beta-galactosidase. Basically, the enzyme that cleaves lactose into galactose and glucose is beta-galactosidase, so that's LAC-Z. So, in this case, this is what the cell wants. If there's lactose outside of the cell, it's going to get in through the LAC-Y permease, and then there's beta-galactosidase in there that's going to cleave lactose into galactose and glucose, which is what the cell wants, because then the glucose can be shuttled into glucose metabolism or glycolysis. So this really only happens when there's high levels of this beta-galactosidase, LAC-Z, inside the cell. Remember, everything is dependent on concentration. So when there's high levels of beta-galactosidase, then you can do this cleavage. But on the other hand, if there are low levels of beta-galactosidase, or LAC-Z, then lactose is converted into another molecule called allolactose. So it's not broken down yet. And then this conversion to allolactose becomes important later on. We'll talk about that. Just remember that for a bit. And so without having enough lac-Y, the permease, to get lactose in, or lac-Z, then the catabolic energy of lactose is then unavailable to the cell. So you can't take advantage of lactose as a carbon source without lac-Z or lac-Y. Questions? You guys take that in. Okay, so remember, this is an operon, which means that an operon is basically you have a DNA sequence that will encode different genes. So the lac-Z, lac-Y, and lac-A operon, basically each one of these are their own separate transcriptional units. So their own genes that can be prescribed or transcribed, but, and they each have their, and these ones have a promoter for the lac-Z, Y, and A, for the actual lac operon. The two that you'll have to know are lac-Z and lac-Y. We're not going to talk about lac-A a lot. But one thing you should know, and that I'm adding to this, is at the very beginning, there's something called lac-I, which is the regulatory sequence that encodes the lac-I repressor. Lac-I is a repressor protein that will repress transcription of the lac-ZYA. So since these are all involved in lactose utilization, or the breakdown of lactose, they all have their own promoter. So if there's a regulatory protein that's bound to this, that allows the promoter to, the RNA polymerase to bind the promoter, then you get transcription of all of these, and you get the proteins. If the lac repressor is active, then you stop the breakdown of lactose. So you don't get transcription of these genes. So lac-I, you have to know, and it's on its own promoter, it has its own promoter, so it's its own transcriptional unit. So the function of lac-A in lactose catabolism is actually widely unknown still, so that's why we're not going to talk about it. But we're going to focus on Z and Y. So if there isn't lactose in the cell, remember, everything is dependent on concentration. So if there is no lactose in the cell, then this lac operon has no need to be, transcribed, and you don't need these proteins to be made. And so the main reason is because the lac operon is repressed by the lac-I repressor. Remember, it's situated upstream of the lac operon for that reason, so it can repress, and it's transcribed by a completely different promoter. Okay. Questions so far?
So is the lac-Z what makes the like lactose usable in the cell?
LAC-Z is the enzyme that breaks down lactose. So if you are, transcribing LAC-Z, then you are going to make beta-galactosidase, and that is only going to be transcribed when there's lactose in the cell. So lactose is usable when LAC-Z and LAC-Y are present in the cell. Yeah, that's why without them, then you can't break them down. But there's also a mechanism in place to stop the transcription of these so the cell doesn't make them. Remember, the cells are going to make unneeded proteins. If there's no lactose, it's not going to want to make beta-galactosidase or the lactose permease. So, you know, in the absence of lactose, they're repressed. In the presence of lactose, they're activated. So how does the LAC-I repressor repress the LAC operon? Well, LAC-I is the repressor protein, and that is, when it's transcribed and it's made into a protein, the repressor basically binds as a tetramer to the operator region. So there's these two operator regions, and so it binds as a tetramer, which means that it has 4 parts. So it's 4 molecules, it binds to this region, and it basically prevents this from being opened for transcription of these, the LAC operon. So instead, the tetramer binds those both operators and it loops the DNA, and it physically causes this loop where it prevents the RNA polymerase from binding here and causing transcription of the LAC operon, LAC-Z and Y and A. So this is what happens when lactose is absent. Once again, why make the genes that break down lactose and the proteins that break down lactose if lactose isn't around? So in the absence of lactose, there's going to be repression, and this is how it works. The LAC-I is the operon, or the gene that the repressor protein in this case binds to. So now we're going to go back right here to where I told you that if there is some beta-galactosidase, but not much, if it's not high, if it's low levels, then it, instead of breaking down lactose into galactose and glucose, it makes another enzyme called allolactose. Allolactose is important because it's actually an inducer. So when there is lactose in the cell, but there's low levels of LAC-Z, or beta-galactosidase, then the beta-galactosidase can cleave and rearrange the lactose to make the inducer, which is allolactose. This is the ligand that is able to bind the repressor, the LAC-I repressor, and therefore it reduces the affinity of the repressor on LAC-I, and then transcription of LAC-Z, Y, and A occurs. So then the cell knows to make more beta-galactosidase because it needs it. So it induces the expression of the LAC operon. So when there is lactose, there's low levels of beta-galactosidase, the enzyme that cleaves lactose, the beta-galactosidase then just transforms it into allolactose, which is then an inducer that is made to bind the repressor falls off, and transcription of the LAC operon can occur, and the cell has now more beta-galactosidase. Does that make sense? Any questions so far? All right. So usually now we're going to think about this, not so detailed mechanistically. We're going to think about this in the environment. In the environment for these bacterial cells, usually in the environment there's more than one carbon source. So the next question the cell asks is, which carbon source should I use? Or can it just use all of them at the same time? Or just does it use one at a time? So as I've said before, the enzymes for glucose catabolism, the breakdown of glucose, they're always present, they're constitutive, and those enzymes are produced at high levels because glucose is the favored carbon source that's providing the quickest source of energy because it can get right into glycolysis. Glycolysis, you make a lot of ATP. So many carbohydrates like lactose have to be converted into glucose before they can be used in glycolysis, and that's not necessarily ideal. So what these cells do is they use this phenomenon called catabolite repression, where a more favorable catabolite, or glucose, will prevent the expression of operons that enable the catabolism of another less favorable carbon source like lactose. So in this graph, we're looking at optical density, so basically bacterial growth. On the y-axis, we're looking at how quickly bacteria are able to grow based on the carbon source that is in there. And this is over time. So optical density measures bacterial growth. The lower it is, the slower they're growing. The higher it is, the higher they're going. And once again, bacteria need to break down these sugars or carbohydrates in order to obtain energy and grow. So utilization of these carbohydrates correlates to their ability to grow. And so both glucose and lactose are present in this media that they're growing in. And so the cell at first grows just fine. And at this point, it's using the glucose, and then it runs out of glucose. And so there's a lag, which means that the cells completely stop growing right here. And then they eventually start growing again, and they're utilizing lactose. And then once the lactose is used up, then they stop growing. So can anybody tell me what's happening in this time frame where they stop growing? Why do they stop growing? Yeah.
Because they need to change their genes to be able to metabolize the lactose.
Exactly. They're literally retooling their genetic program in order to now be able to turn on expression of the lac operon genes so that you can utilize lactose. And so this is called dioxic growth, where you have this biphasic growth curve of a culture growing on two carbon sources. All right. Questions so far? See here. Okay, so we'll just go through this and then we'll stop after this. But just to put this all together, one of the ways that this happens is when you have glucose present, it prevents lactose from getting into the cell. And so if the lactose can't get into the cell, then the lac operon can't be induced. So one way is when there's glucose that's being transported into the cell by another membrane protein, then this inhibits lac-Y, which is the permease that allows lactose to get into the cell. So this is called inducer exclusion. Because if lactose can't enter the cell, then the operon cannot be induced. It's excluded. So that's just another term. If glucose is absent, then LAC-Y can be made, the permease that allows lactose in, and then you have lactose in the cell, and it can be broken down. Okay, does that make sense? Glucose can repress the lac operon, and this is called inducer exclusion. And it does so by inhibiting the permease this time, the process of getting lactose into the cell. Questions? Okay, we'll stop here, and we'll continue on Friday.
Okay.